Hi, my name is Hopi Hoekstra and I'm a professor at Harvard University. Today what I'm excited to do is to tell you about the field of evolutionary genetics and in particular the genetic basis of evolutionary change. I'm going to tell you two stories, one about morphology and one about behavior. So here's the outline of the three segments of my presentation. So what I'm going to do now is give you an introduction and introduce you to some of the long-standing questions in the genetic basis of adaptation and give you a sense of how we're addressing these questions. And then the second segment in particular, I'll tell you a story about how we're tracking down the genes and developmental mechanisms involved in camouflaging color differences between two species of wild mice. And in the third segment, I'll tell you about how we're using very similar approaches to track down genes involved in burrowing behavior differences in these same mice. So I want to start today by talking about Darwin, because in 2009 we had a number of celebrations celebrating everything that Darwin knew on his 200th birthday and on the 150th anniversary of his magnum opus on the origin of species. Now Darwin certainly knew a lot about evolutionary change, but there is one thing that he didn't get quite right, and that is the mechanism of evolutionary change, or the genetic nuts and bolts about how organisms adapt to their environment. Now Darwin knew traits were inherited, he knew that offspring resembled their parents, but he didn't know how. And this maybe isn't surprising because during his time, of course, we didn't know about DNA or genes, much less the whole genome. And that's really what I want to focus on today, is this mechanism of how changes in genes actually produce variation in phenotypes on which natural selection can act. So I'm going to start by telling you a brief anecdote that links Darwin to a second great discovery, and that is the discovery of DNA. So what I'm showing you in this next slide is Darwin's last publication. Now I don't expect you to read it, but I just want you to appreciate the fact that you're looking at his last publication. It was published in 1882, just 2 weeks before he died, in a prestigious journal called Nature. The title of this article is called On the Dispersal of Freshwater Bivalves. And what this really is a report of the finding of a freshwater beetle clamped to its leg was a freshwater clam, or a cockle. So why, you may be wondering, was this published in such a prestigious journal even 100 years ago? Well, this actually resolved this great debate about why freshwater cockles were so similar among disjunct lakes in the British Midlands. One hypothesis was that these cockles could migrate from lake to lake, thereby homogenizing the populations and thereby making them very similar in size and shape. But the big question always was, well, how do they get from lake to lake if they can't cross terrestrial habitat? Well, here was a mechanism. They could hitchhike by attaching to things that could fly or traverse this terrestrial habitat, in this case by clamping to the leg of a beetle. But that actually isn't the point of telling you the story. The point of telling you this is to mention that Darwin was sent this beetle with the cockle clamped to its leg by a young shoemaker who was working in the British Midlands, who was an amateur naturalist. And his name was Walter Drawbridge Crick. Now this name should ring a bell because his grandson was the one with his colleague Jim Watson that made this second great discovery, that is the discovery of the three-dimensional structure of DNA. And it's in this DNA text that we find even more evidence for Darwin's great theory, that is our 3 billion year existence, the shared evolutionary history of all living organisms, and the subject of what I want to talk about today, and that is the mechanistic basis for evolutionary change. So like Darwin, one of the big questions in evolutionary biology today is what gives rise to this amazing diversity? How is variation generated and maintained in natural populations? But thanks to Watson and Crick, we can look for that answer in the genetic code. So the big question that we're focusing on is, what is the genetic basis of fitness-related traits? By fitness-related traits, I mean traits that improve the probability of survival or reproduction of organisms in natural populations. So finding the genetic changes or the precise DNA changes that contribute to variations either between populations or between species is a fun endeavor, and we certainly can learn things about the mechanistic aspects of evolutionary change, like how do changes in genes actually produce changes in phenotype. but I'd like to argue that we can actually learn even more about the evolutionary process. So what can finding genes tell us about how evolution works? Well, there are a number of long-standing questions that I think we're just now starting to be able to answer because we're armed with molecular biology and the ability to link genotype and phenotype. So I'm just going to list a few of these big questions. So for example, how does evolution proceed? Is it through many small changes, many small mutations, each that have a small effect on a trait, or can evolution take big leaps? That is, can mutations have large effects that are beneficial? We also want to know about the dominance of these mutations. So do adaptive alleles or mutations that appear, do they tend to be dominant or recessive? So J.B.S. Haldane, one of the founders of population genetics, argued that adaptive mutations tend to be dominant because when they first appear, they're visible to selection and then can quickly spread through the population, compared to a recessive mutation, which would have to build up enough in number in a population to be contained in the same individual and that recessive trait then expressed. We also want to know how do these mutations interact? So if multiple mutations are responsible for changing a phenotype, do they interact in a complex way, or does each mutation additively affect that trait? We also want to know where these mutations, these beneficial mutations, are. Do they occur in the protein itself, for example, amino acid changes that affect the structure and function of that protein? Or do they occur in what we call non-coding DNA, which affects the regulation, let's say the timing or place of expression of that protein? And then we want to know where these mutations come from. So for example, if there is a change in the environment, do we have to wait around for new mutations to appear in that population? Or are there these mutations maybe at low frequency in the population already that are pre-existing that can be selected on almost immediately? And then finally, if we find mutations in one population that are responsible for an adaptive trait, and we have a similar trait evolve in another population, is it the same mutations and same genes that are responsible for those convergent traits? Now, importantly, all of these questions that I've listed don't have simple yes or no answers. In fact, we're more interested in the frequency, whether, for example, more often beneficial mutations occur in regulatory regions versus structural regions. But even more importantly than that, we want to know why. Why in some cases do we see protein coding changes, in other cases we see regulatory changes. Now these, I would argue, are still largely open questions, but questions that we can start to answer by making the connection between genotype and phenotype. So the context in which we're studying the genetic basis of adaptation looks like this. That is, we're trying to make the connection between environment and phenotype. In other words, trying to implicate a role for natural selection in driving that phenotypic variation. That is, suggesting that the phenotypic differences affect fitness. But we also want to understand the genes underlying that phenotypic variation, and not just what those genes are, but how those genes, through, let's say, development, actually produce the differences in variation. And then once we make these links, we'll have a much more complete picture of the adaptive process. I think this is where things can get really fun because we can go back out in the wild and ask how traits evolved in nature. So to make these links between environment, phenotype, and genotype, my lab group is studying one particular group of wild mice, commonly referred to as deer mice, or mice in the genus Peromyscus. Now these are the most abundant mammal in North America. And the reason we study them is because first, they're found in almost every habitat type. So from the top of the Rocky Mountains, out to the shores of Maine, to the plains of Kansas, to the deserts of Arizona. So they're very widespread in their distribution, and because they live in all sorts of different habitat types, there's a lot of opportunity for local adaptation. So in addition to all the variation that we find in the wild, they also can be treated much like laboratory mice. That is, we can bring them into controlled laboratory environments. They breed in the lab just like laboratory mice, and we can do controlled experiments. And finally, while we're still behind traditional model organisms, my group, as well as others, are building a series of genetic and genomic tools that are going to be useful in trying to make these connections between genotype and phenotype. But I would argue one of the main reasons we're studying these mice is because we have this amazing literature of natural history studies on their ecology. That is, these mice have been studied for nearly a century by natural historians who have described morphological, physiological, reproductive, and behavioral variation in natural populations. Just to give you a sense of how these mice vary, here are just a number of traits that I picked out of the literature that describe traits that have been studied and traits that vary either between populations or between species of Peromyscus species. So they vary in body size, tail length, foot size, color patterning, testes size, sperm morphology, etc. They vary in morphological traits, physiological traits, and behavioral traits. So using these mice, we're trying to make these connections between genotype and phenotype. In the next two segments of my presentation, I'm going to focus on two of these traits. One morphological trait, color patterning, and a second trait, burrowing behavior. So in the second part of my presentation, what I'd like to do is focus on a morphological trait, and in particular, camouflaging color differences between subspecies of Peromyscus polyanotus, both to understand the ultimate reasons why these color differences evolved, as well as the proximate mechanisms or the underlying genetics contributing to these differences in camouflaging color. And for the third part, we'll switch gears and focus now using very similar approaches, but instead of studying a morphological trait, we've substituted in a behavior where we're taking advantage of these dramatic differences in burrowing behavior, either species that build these large burrows versus those that build small burrows, to try to understand how genes can affect behavioral variation in natural populations. So I hope to have gotten you excited about biology in the sense that we're at this amazing time where we can use approaches like Darwin first did, that is, studies of natural history, observation, and experiment in the wild, but combine that with studies of modern-day molecular genetics to try to understand the genetic basis of what Darwin referred to as that perfection of structure and co-adaptation, which most justly excites our admiration. So thank you very much for your attention, and I hope you'll join me for the next two segments where I'll tell you about more detailed studies from my laboratory group, trying to connect genes and phenotype for both morphological and behavioral traits. Thank you.